Hello guys, today we will talk about genetic polymorphism. Genetic polymorphism refers to the genetic variation at the level of DNA sequence within a population. It involves differences in the nucleotide sequence of a particular gene in the genomic region among individuals. There are four main types of genetic polymorphism: single nucleotide polymorphism, insertion and deletion polymorphism, copy number variations, and variable number of tandem repeats. Single nucleotide polymorphism or SNPs. Single nucleotide polymorphism are the most common type of genetic variation involving a single nucleotide change at a specific position in the DNA sequence. They occur in both coding and non-coding region of the genome and can influence traits and disease susceptibility. As the structure shows that all the three samples are same, but they have got different nucleotides at a specific location. Number two, insertion and deletion polymorphism or indels. Insertion and deletion polymorphism are genetic variation characterized by the insertion or deletion of one or more nucleotide within a DNA sequence. They contribute to genetic diversity within a population and are valuable markers for genetic studies. Insertion and deletion polymorphism are associated with susceptibility to disease, drug response, and other phenotypic traits. In insertion and deletion polymorphism, the new traits are added to the DNA sequence from as a result of mutation and recombinant DNA technology. Copy number variation are CNVs. Copy number variations are structural variation in the genome characterized by differences in the number of copies of a particular DNA segment compared to a reference genome. CNVs can range in size from kilobases to megabases and involve duplication, deletion, multiplication of DNA segments. Copy number variations are based on the number of copies that an individual gets from parents. Number 4. Tandem repeats polymorphism. Tandem repeat polymorphism consists of short DNA sequences that are repeated consecutively in the genome. The number of repeats can vary between individuals, leading to polymorphism. There are two main types of tandem repeats polymorphism: VNTRs and STRs, variable number of tandem repeats and short number of tandem repeats. VNTRs are longer, means they consist of 10 to 100 base pairs with a variable number of copies of a repeating sequence. They are often used in the DNA fingerprinting and population genetic studies due to their high variability. Short number of tandem repeats are also known as microsatellites. These tandem repeats consist of short sequences that are usually 1 to 6 base pairs repeated multiple times. They are used in genetic analysis for forensic identification, paternity testing, and population genetics. Tandem repeat polymorphism refers to the number of copies that are repeated in the DNA sequence. That's all for today. See you in the next video. Goodbye.